All right, let's do a real quick recap. Tell me, what is this?
Homologous pair.
Oh, wait, that thing.
Nice, DJ. Homologous pair. That's all right. I'll take homologous. Homologous pair. Sure. Oh, there. Okay. How about our result? What's over here? What?
Gamete.
I heard it. Gametes. And in the middle, how do we get from homologous pair to gametes?
Crossing over.
Crossover. I almost said homologous. All right. In the Q column, I need you to write metaphase 1. Metaphase 1. Because we already wrote meiosis 1, right? We just need to write metaphase 1. Right?
We did write meiosis 1.
Yep, so we just need to write metaphase 1. We don't need to write meiosis 1 again. Metaphase 1. This is the second phase of meiosis 1. Second phase of meiosis 1. Crossover is complete. It's done. It already happened. When did it happen?
Yesterday.
First phase is what?
Prophase.
Prophase.
Metaphase.
1. There we go. We have to be specific because prophase happened in mitosis. Prophase 1 happens in meiosis 1. You have to add the 1. So, crossover is complete. The homologous chromosome pairs move to the middle. They line up like the equator. And the centrioles and centrosomes are at the poles. The spindle fibers are controlling everything like our marionette puppet. Okay? They're getting ready. So prophase is like the prep. Metaphase is, all right, we're lined up, we're ready now. What's crossover?
Where the two genes cross over.
Okay, how else can we use? How else can we define it without using the word in the definition?
Is it part of a prophase?
Swaps genetic material. I like it. There we go.
Two.
Okay.
Homologous.
3D prefix. 3D prefix. 3D prefix. So the processes of mitosis and meiosis and all of it were named after some Greek words. Okay, some old Greek words. I know, and from what I'm hearing, you guys have been learning prefixes in Coach Gunner's class too.
Yes.
So we're going to do more with prefixes. All the prefixes in meiosis and mitosis have these meanings. So pro means before or in front of. Meta means after or middle. Ana means again or return. And telo means end or completion. What I want you to do is I want you to tell me, how do these prefixes of the phrase fit with that particular phase of cell division? And here's what I will allow. Because it's set up like this, you don't necessarily have to write it in full and complete sentences. You can write pro dash and then give me your explanation. Okay? So how does pro fit prophase? What do we know about prophase that makes pro fit
It's the first.
that that meaning? Anna?
I just kind of said like prophase is like the beginning that happens before everything.
Yeah, it's like the beginning and it happens before everything. It's the first, right?
Is that okay how I said it?
Yeah,
Okay, what?
I like that.
Beginning.
It's the beginning.
It's in front.
It's the first stage. Hey, Aiden, how about meta?
Meta, active.
What?
The meaning, right?
Middle.
Oh, middle.
Kate?
The second phase, it says in the middle of it.
It's in the middle of going
Metaphase?
through all of the phases.
It is in the middle of all of the phases, but why else could meta mean or fit that particular phase?
First middle.
Jordan,
Because it comes after the first phase.
it comes after the first phase.
In the
DJ,
middle of the nucleus.
in the middle of the nucleus? What's in the middle of the nucleus?
Oh no.
The chromosomes line up in the middle, right?
Homologous.
What about Anna? This one's a little bit more difficult. And makes your thing very difficult.
What is it? Anna?
Anna. Why does Anna fit anaphase? Why does this mean, again a return, fit anaphase? Aiden?
Doesn't it should be like in the middle of hold on. We'll go back.
Yeah. Let's figure out what's happening in anaphase that makes, again, a return fit anaphase.
Circle.
Hey, come on.
Happens twice. Hey, giggles.
So it's like taking it apart of, but then they like move back together eventually.
Take apart but move back together.
Eventually.
Is that what's happening in anaphase? Do they come back together in anaphase?
I don't think
No.
so.
Well, they're.
They go
So
apart.
I think it's mainly because in anaphase they're like splitting apart so they can turn into the separate cells and it can like kind of start over again.
They're starting to return back to their original state. Yeah. When they're starting, the chromosomes are separating, it's because they're trying to return back to their original state.
It's when the chromosomes move to the opposite poles.
Yeah, they're starting to move to the opposite poles.
Doesn't tell us.
So, again, remember, pro, it's, all right, we're prepping everything. Meta, we're ready. Let's go. Anna, it actually starts that return process. So then telo, endocompletion, how does endocompletion fit telophase? It's a what?
End of the cycle.
It's the end of the cycle, it's the last phase, what else?
The cells that were made are now like
Game over.
maturing.
Yeah.
Game over.
And they're
We
like
have-
completed.
We have 2 complete daughter cells, right? We have diploid or the 2 daughter cells for meiosis 2 ready to go to start again.
Oh, so that would be like.
Yeah.
Reloading
Make sure you get it
shells,
written down.
loading shells in the truck, and that one would be on the back, and that one would be firing, and that would be the dead bird.
Let's not use gun references at school.
Well, it just helps me.
You're on camera, bro.
Great. Let's just follow along with us. Okay. Participate in discussion. Okay. Okay. All right. I'm going to move forward. Let's write anaphase 1 in the Q column. The third phase of meiosis 1. Anaphase 1, third phase of meiosis 1. Anaphase 1 is just like anaphase, where the cell elongates. The centrioles and centrosomes are still connected by spindle fibers to the chromosomes, and they're pulling the chromosomes apart. The difference is we have homologous chromosomes that are moving. And the sister chromatids get to stay together. Okay? The homologous chromosome pairs are split apart from each other, but sister chromatids stay together this time in anaphase 1. All right. Remember, we want to pick out what's important, not write it word for word. Okay? Oh. What remains together and what separates during anaphase 1?
Chromosome separates and the chromatid remains together.
All right.
The chromatid.
Chromatids stay together, yeah. Chromosomes separate, chromatids stay together. All right. Telophase 1. I want you to notice that figure 8 shape stays the same. Okay? That cleavage furrow still shows up because that's part of cytokinesis where the cytoplasm separates. That's what causes that figure 8 shape. The difference here is now we have 4 sets of chromosomes. Okay? How many chromosomes are in a gamete? 23. We have 4 sets of that. So what does that mean? What's 4 times 23?
42.
Times 23 times 4?
96.
92. 92. So in this one cell, before it undergoes cytokinesis, we have 92 chromosomes. Okay? At the end of come back. At the end of cytokinesis, Aiden, I need you paying attention.
Okay.
At the end of cytokinesis, we have 2 daughter cells with 92 chromosomes split between them. So that's going to be 46 in each one. But we're not done splitting.
TJ, do not talk to me.
Are we done with meiosis?
Yeah.
Oh, no.
No. Why?
Because it didn't say last phase.
It didn't say last phase. Why else?
Don't they have to split into 4? Not three?
Because we end up with 4, right? We're not at 4 yet. We're at 2. So we have what's called interkinesis. Yeah, interkinesis in the Q column. Kinesis means separation. So remember interphase on mitosis, that was the in-between stage. Okay? Interkinesis is an in-between stage of meiosis. Okay? So our daughter cells have separated, the cytoplasm has separated, but now we have 2 daughter cells resting, getting ready for another separation. But now there's no while the chromosomes unwound back to chromatin, it's all loose and stringy, it's not going to replicate now. It just chills. Okay? When we started meiosis, how many cells did we start with?
5.
1. At interkinesis, how many cells are we starting with? 2.
Yeah, I had 8 shots.
All right. In the Q column, I want you to write meiosis 2. No, my bad. Hey, let me see yours. Eyeballs in your balls. Okay? On this quick IMD, you know the new cutouts that you added yesterday? Here's what we're going to do. Okay? On the top part, you're going to go in and write a 1 after each of the labels. Okay? Because we're going to make this meiosis 1 and meiosis 2. So the top one is going to be meiosis 1 at 1. Instead of interphase, it needs to be what? Inter what?
Interkinesis.
Interkinesis. Thank you, Josh. Okay. So then here's what we're going to do. Just like on mitosis, where we drew on the top part here, we're going to draw, but not right now. Okay? I'll tell you when. On the back part, not this, or eyeballs and earballs, not this, right here is where we're going to write this. Okay? So not here. We're not writing here. We're writing here. Okay? Okay. Everybody understand? Okay. So let's write down a little bit of information. In which phase is the cytoplasm divided by cytokinesis? Yes, that's a good answer.
There's another cut.
Telophase 1. Thank you, Anna. In which phase does the nuclear envelope begin to disappear?
Yesterday.
In which phase does the nuclear envelope begin to disappear? Yeah. You want to talk? Give me an answer.
So, what was the question? And I'll answer that. What was the question?
For the third time, in which phase does the nuclear envelope begin to disappear? Do you agree with Aiden?
No.
Which one is it?
Meta.
The nuclear envelope disappears in metaphase?
No.
No.
Telophase.
All right. So, the nuclear envelope begins to disappear when? Prophase. 1. We have to be sure and include that number, the 1, afterwards, because we've got 3 prophases that we're going over. We have to be clear in our vocab. I know, but we did one during mitosis also, right? Okay. Which phase do the chromosomes line up in the middle of the cell?
Metaphase.
Metaphase.
1.
1. All right, Jordan. Which phase do the chromatids remain together instead of separated? Anaphase 1. All right.
I think this last one is
And then with interkinesis, you should be able to write, that on interkinesis and anything else that you can think of. Right? Right. All right. Let's move forward. Hey, Venn diagram. You can add this to your notes for extra, but we're moving forward. Okay? Okay. Let's talk meiosis 2. Meiosis 2 in the Q column. Prophase 2 underneath that. Meiosis 2, prophase 2. Meiosis 2, prophase 2. Now everything looks more like mitosis. Except it's twice over. We start with 2 daughter cells. That's our beginning state. Chromosomes are still condensed. Nuclear envelope still breaks down. Centrioles and centrosomes still start to move away from each other and towards the poles, and spindle fibers still begin to form to be able to control those chromosomes. But now it's just 2.
not those.
Start with 2 daughter cells. Okay, these are the same 2 that we had in interkinesis. They're not brand new from somewhere else. We're just continuing the process, right? Metaphase 2. Prophase 2 is preparing. Metaphase 2 is, all right, we're ready. Centrioles, centrosomes are at the poles, controlling the chromosomes through the spindle fibers. Chromosomes are lined up on the equator of the cell. But now look at our chromosomes. How do they look?
They're flipped.
They're flipped?
Like crossed over.
They've crossed over, right? Yeah. They don't look the same at any point.
That's what I was going for.
I understood you. Because in meiosis 1, our homologous pairs went through crossover, so now our chromosomes all look different.
What does that say?
Centromeres, remember centromeres are the centered, our diagram showed it like a button, but it's not really like a button, it's just where the spindle fibers connect.
A little ball in the middle.
Yeah, where it all combines at the middle, the center of the chromosome. Alright. Anaphase 2. Third phase of meiosis 2. Okay. The cells elongate, because we have two. Both of them elongate. And the sister chromatids now get split apart. So we have individual chromosomes attached to the spindle fibers that are pulled. Thursday. What is the answer for that? What do you see? What does it say? No, but is that what we see right now? We're not worried about identical twins right now. What do we see here?
Yeah, we're not with the genus.
Because even with identical twins, this is still going to show up like this. Okay. Alright. Are we there?
Oh, hold on. Never mind.
You good?
No, I got
No.
no computer.
Oh, okay. Here. Telophase 2. Last phase of meiosis. Not just the last phase of meiosis 2, it's the last phase of meiosis. Okay? Chromosomes reach the poles. We already have our two daughter cells, but now cytokinesis is separating the cytoplasm on each of them. We're going to end up with 4 gametes. What are gametes? 4 daughter cells, but they're specifically our gametes. Okay? What are gametes? Huh? They're the reproductive cells, right? They're germ cells. They're going to be genetically different from each other and genetically different from the initial parent cell. None of these are alike. Nuclear envelope starts to reform again. The end result is 4 haploid daughter cells, gametes. Okay? Okay.
Deal.
I think it's a quick IMB mix, yeah?
Yeah, for sure.
Yeah. Maybe it was bathroom.
Yeah.
Can you
Alright. You good, Cub?
No. His blood is not over it.
Huh? What?
Alright, you got it.
Alright, good. Yeah, quick IMB. Hey, the second flap. So yesterday's cutouts. We had the top flap and the bottom flap. Let's go to the bottom flap. And the back side of the bottom flap. Okay? That's where we're gonna write these. So underneath it on the your actual paper or the back side of the flap.
I'm gonna do underneath it.
Okay. Alright. Which phase do the chromosomes line up in the middle of the cell?
Metaphase.
Metaphase.
1.
2. We're on 2 now. We're talking meiosis 2. Metaphase 2. So, since we have that bottom flap, do we need to change the labels on the flaps?
No.
To add 2?
No.
Prophase 2, metaphase 2? Yeah.
Wait, so what do we do?
The bottom flaps, make sure you are writing 2 after each stage. Okay? We've already written 1 on the top flaps. Now we have to write 2 on the bottom flaps. And then you're going to write underneath it the information on the slide. What does it say?
In which phase? Oh, it's metaphase 2.
So let's try that again. Which phase do the chromosomes line up in the middle of the cell?
Metaphase 2.
Metaphase 2. Are you seeing the trend here?
Yep.
It doesn't matter which metaphase it is. Metaphase, metaphase 1, metaphase 2. The chromosomes always line up in the middle. How about, in which phase do the chromosomes move toward the poles? Anaphase. Anaphase what? 2. Anaphase 2. Again, do you see the trend? Every single anaphase in cell division. Anaphase, anaphase 1, anaphase 2. Chromosomes move toward the poles.
I didn't even know I stepped on it. What happened?
How about, in which phase has the cell divided into 4 haploid cells? Jordan.
Metaphase 2.
Telophase 2. End result is going to be telophase. And in which phase do the chromosomes condense? Prophase 2. Again, do you see that trend? Prophase, prophase 1, prophase 2, chromosomes condense. They're getting ready. Okay? Make sure you're writing this down underneath that bottom flap. The last one is still going to be, on the bottom flap, it's still going to be interphase. Or X'd out, ignored. We're not really doing anything with it. Okay. Summary. Let's bring it all together. Okay? Promise, we're almost done.
Yeah, we have 7.
Trust the process here. Okay? Summary. Meiosis is a type of cell division that reduces the number of chromosomes in half. Okay? And for gametes like sperm and ova. Here's what's going to happen. I need you to write this down, okay? Pick out the important parts. Don't write it word for word necessarily. And then you're going to make room and you're going to draw this. The bigger you draw it in your notes, the easier it is to write the labels and so you can understand it. We're going to go over this first, okay? And then we're going to go over this in a little bit more depth and hopefully it all makes more sense. Yeah.
I'm not good at drawing.
It's circles and lines. Yeah.
That's pretty hard to do. I was not saying I'm on.
I am
Here,
artist.
no. In mitosis, yes. Okay, so let's do that again. Meiosis. It's a form of cell division that results in half the number of chromosomes in the gametes. It allows the species to maintain the same number of chromosomes from generation to generation. And it results in genetic variation being passed generation to generation.
I'm not LeBron James. Does the drawing get us points?
Yeah.
I'm not in it. Still sports.
All right. Again, type of cell division results in half the number of chromosomes in the gametes. It allows for the species to have the same number of chromosomes in every generation moving forward. It also allows for genetic diversity in each generation moving forward.
DNA.
So what we see in this diagram is how the chromosomes are replicated and then cut in half. We're going to go through it step by step, and I'm going to explain it to make this make more sense. But I'm going to give you a chance to draw it, okay? What slide number is this? 41? Yeah, so I'm 41. Make sure you're drawing that.
Do we have to write it all out?
Yeah, the more you label, the more sense it makes. And then we'll add even more labels.
Homologous.
Drop it.
Homologous.
What?
Say it.
Alright.
Homologous.
What in the world?
Homologous.
You're on camera.
Homologous.
No. They can see me?
What do you say?
What is he doing?
Where's the camera?
It's on the
What
lamp.
do you do?
Okay.
What did you do?
Alright. How many chromosomes do you have in your cells?
23.
Josh, say that again. 46. Alright,
I was saying 92.
up here. Pay attention. Okay? Let's try that again. Josh, we have how many chromosomes in our cells?
46.
46. So, in the process of replicating, or in the process of meiosis, we have to replicate our DNA, right? If we're going to pass on our DNA to the next generation, to offspring, we have to give them some of our DNA, correct? Because as an offspring, you get half of your DNA from this parent, half of your DNA from that parent, right?
Yes.
Right? Okay. So, bless you. If we have 46, we have to keep 46 chromosomes in our cells. How are we going to give 23 to offspring and keep 46?
We create more.
Well,
Well, I mean, matter cannot be created nor destroyed, only transformed. But yes, we create more. We have to make copies. So, through DNA replication, this is what's happening in interphase, right? Our DNA, when it's unwound chromatin, makes the copies. Yes? Yes, it makes the copies. So now, huh? Hey, this is where things get hard to understand. Mitosis and meiosis can be difficult for some people. It helps if you're paying attention.
I got one.
Okay.
I wrote it all down.
Okay. So during interphase, that's when replication occurs. So if you wrote down DNA replication, you can also write interphase here. So we know what and when is happening at which phase, right? And we end up with 92 homologous chromosomes
I thought that was just
going
animals.
into prophase 1.
I thought that was just animals.
You are an animal. Humans are animals.
Oh, okay.
Humans are animals.
Actually, BT permanent.
Okay. Okay. So prophase 1, we have 92 homologous chromosomes. Then we go through metaphase, anaphase, telophase, and end up at our two daughter cells in interkinesis. And each of those two daughter cells in interkinesis have 46 chromosomes apiece. But we're not done splitting, right? Because we're only passing on
Stuff you don't have.
We're only passing on half. We're not passing on 46, we're passing on half. Because as humans, there can only be 46. And if you get half from this parent and half from this parent, then you can only give half of 46, which is 23.
So then,
What happens if somebody gets a little bit more than 46?
We can discuss that in a minute.
that was,
Okay? So then, Here, if you drew this, next to here, you can write interkinesis. Because that's where we start meiosis 2, or in between meiosis 1 and meiosis 2 is interkinesis. Then the rest of the phases of meiosis 2 occur for us to end up with our 4 gametes, or haploid gametes. And how many chromosomes are each of our haploid gametes going to have? 23. That's how we.
She broke.
I gotta give you that one. That's how we pass along our chromosomes. Okay? Because our haploid gametes have half of what our cells start with. Does this make more sense? Yeah? Yeah?
Heck yeah.
Yes? No?
You have to speak English.
Okay. This 3D thinking, we're gonna skip it, because I don't like it. I don't see how it helps understand the standard. I don't see how, yeah, I don't like it. Alright. Draw a Venn diagram.
Wait,
two circles.
Yep, that's the 2 overlapping circles. Make it big enough so you can actually write in it. Let's draw a Venn diagram. Oh, we're going to compare mitosis and meiosis. Yeah, I guess. The important thing is the information.
Figure out why.
Alright. 5, 4, 3, 2, 1. So tell me, what does meiosis and mitosis have in common?
Type of cell.
What?
Type of cell.
They're both types of cell division. What else? Nick, give me one.
Different
It replicates DNA.
Oh, replicate.
They both replicate DNA. DJ.
Mitosis is like
It's like what?
the haploid or whatever it's called.
Mitosis ends with haploids?
No, the diploid and haploid. Or whatever.
Okay, tell which one's which?
Mitosis is diploid.
Mitosis is?
Haploid, or however you say it. Whatever you say.
You
Come on.
got this, DJ.
Come on, DJ.
Come on, DJ.
Haploid.
Come on.
I don't know.
Try harder.
Computer, buddy.
It's on the board too.
Use your context clues.
Diploid. Mitosis ends in diploid cells, which means meiosis ends in haploid. Haploid.
Oh, I got mixed up.
Okay.
Here you go, DJ.
Give me another thing they have in common.
Replicates.
Creates.
Nate already told us that one.
Creates new cells.
Creates new cells. Now we know matter cannot be created nor destroyed, only transformed. But yeah, we're gonna go with creates new cells.
Oh, one more.
What do you got?
A type of cell division.
Jackson already told us that one. Alright. Give us something different between the two. Brayden, give us a difference.
Oh my gosh.
I have one.
Okay, Sav, what do you got?
Well, like, meiosis is, like, really only through, like, meiosis, is only through, like, the sex cells and, like, the gametes.
Yeah, meiosis only happens in the sex cells. What's another name for that? Gametes. It results in gametes. Germ cells. So then mitosis happens where?
Oh, yes. In the nucleus.
In the body.
The body, also known as what kind of cells?
Animals.
Somatic cells. Thank you, Kate.
Oh yeah, not germ cells. It's
Yeah.
germ cells.
Give me another difference.
Meiosis happens in sex organs.
Yeah, we already said that.
Oh.
Literally just said that one. Literally. Steph,
So meiosis has 2 stages.
meiosis has 2 stages. What else, Steph?
And then mitosis only has 1 stage.
Mitosis only has 1 stage. All right.
What's so funny about that?
All right, I'm going to move forward. Hope you guys got all this down.
We did.
Okay. We're going to skip that. And hey, we're at check for understanding. But I need you to go back to your cutouts. To your cutouts. The ones we wrote in today. The cutouts you wrote in today. And go to slide, what slide number is this? Somebody help me out.
It is slide number. Let me say it. Hey, five.
16?
47.
This one?
47.
Oh yeah, I was looking at it. I'm sorry, guys.
This is slide, 25.
25, slide 25. Okay. On your cutouts, we already drew what the cell looked like at each stage of mitosis. There's a reason we relabeled the cutouts today for meiosis. You're going to draw what the cell looks like during each stage of meiosis 1 and meiosis 2. Okay? Remember, in meiosis 1, the difference is the homologous chromosomes. In meiosis 2, the difference is it's twice over. You have 2 cells to draw in each stage in meiosis 2. If you need to use colored pencils so you can tell the difference because you know crossover happened and you need to show crossover, then use colored pencils. If you need to label the different pieces so you understand what's happening, then label the different pieces. Okay? But that's where we're finishing for today. You have the rest of our time to draw this on those cutouts.
When are we grading your
Okay?
notebooks?
Tomorrow. We're gonna grade tomorrow.
After our lab.
All right.
There's no lab.
There's no
All
lab.
right.
What? On their